Hi, my name is Ellen Weinzappel, and I'm a product manager at EpiCypher. In our recent chromatin mapping series, we explained how chromatin mapping data are aligned to the genome to create sequencing tracks. But there are other ways to visualize these data. Here we will discuss how to interpret sequencing heat maps. As a small refresher, let's go over sequencing alignment. Sequencing cut-and-run, cut-and-tag, or ChIP-seq reactions produces millions of short reads that correspond to where your target is located on DNA. Scientists align or map these reads to the matching sequence in the genome. Here I am showing a small segment of the genome as an example, but sequences are mapped genome-wide. Mapping the reads allows you to know the amount of target at each location on DNA. This is called enrichment. Areas with many aligned reads have high enrichment, while areas with fewer reads have low enrichment. Aligned reads are normalized and converted to something called sequencing tracks, which show the average signal at each site in the genome based on the quantity of mapped reads. Genomic regions with more mapped reads generate peaks in sequencing tracks, while areas with low enrichment do not show peaks. These tracks are generated for the entire genome, meaning you can look up any gene you want and see how much of your target is bound. There are many other ways to analyze sequencing data. Sequencing tracks can only show one region at a time. But what if you want to visualize data in a genome-wide manner? This is where heat maps come in. Heat maps visualize target enrichment at a defined set of genomic regions, such as transcription start sites or gene bodies. This example shows enrichment of a histone PTM around the transcription start site, or TSS, of more than 18,000 genes. Note that this heat map is not showing every peak or aligned read. It is only showing data around the transcription start site. If you zoom in, you can see that the heat maps are organized into individual rows. Each row contains data for one gene. The color gradient goes from red to blue, indicating high to low enrichment. For this heat map, the data have been sorted such that genes with high enrichment are at the top of the heat map and genes with the lowest enrichment are at the bottom. And that's it! If you are interested in other informational videos, let us know. Visit epicypher.com and click Contact Us, or email me at ellen@epicypher.com.